So hello guys, hello everybody, my name is Deyvid Amgarten and I'm a master's student in the program Interunities in Bioinformatics at the University of São Paulo, Brazil. I'm guided by Professor Ricardo Vêncio and I'm doing this seminar to the discipline Seminary and Advanced Topics of Bioinformatics designed by Professor Ricardo Vêncio. In this seminar I'm going to talk about viruses and bioinformatics. That are two things that I like and I put them together. And my seminar will be mostly based on a Coursera course that I've taken. It's called Virology 1, How Viruses Work. It was given by Professor Vêncio from the Columbia University. It's a very good one, it's a great one. If you want to know more about virus, you should check on it. It's coursera.org. And I hope you enjoy the seminar, so let's go. This is a summary of my presentation, and I have divided my seminar in three main parts. And the first one, I'm going to talk about the viruses, and I'm going to contextualize you guys what are viruses, how can you define a virus. I'm going to talk about this different concept about virion and virus, what are they, and I'm going to talk about their structure. I'm going to talk about their genome and classification. That are two things that are really related. I'm going to talk about life cycle and these special types of viruses that are called bacteriophages. And I'm throwing you this question already. I'm sure that everyone, this question pop-ups in the mind of everyone. If are viruses alive? So I'm going to explore a little this question. I'm not going to give you the answer. And I've selected the main topics that I think is important to the bioinformatics, because viruses is the object of my study in bioinformatics. So these are the main topics that I think it can influence the bioinformatics you do, the bioinformatics that I do. And the second part, I'm going to talk about metagenomics. And I'm going to contextualize this new era that we are experiencing. The difference between genomes to obtain genomes and metagenomes, the genomic era and the metagenomic era. I'm going to talk about why we are doing metagenomics. What's the purpose of that? I'm going to talk about that, and I'm going to show you some metagenomic projects. Some of them all of them really interesting. And in the end, I'm going to talk about viral metagenomics. That is some metagenomic more specific to virus communities. So it's very important, and that I finished my second part. And the third part, the last one, I'm going to talk about the bioinformatics applied. So after all the things that I'm going to talk to you, I'm going to try to conciliate the bioinformatics with these things. So I'm going to talk about the challenge that we have to study viruses, and the approach you can have to study viral metagenomics, and the challenging tools that we have to study metavirome. This is I'm going to talk about the challenges we are having and show you some of the tools that try to address this problem. So let's go to the part one of it. So this is the part one of my seminar. In this part I'm going to talk about viruses, and I'm going to do an overview of the things that I think is important about viruses to bioinformatics. So it's just an overview, as I said. If you want to know more about viruses, if you want to go deep, you should check on the Coursera course that Professor Vêncio gives, and then there you will have much more knowledge, much deeper knowledge about viruses. Let's start. So the first question that pops up in our mind is, what is a virus? So there are some ancient, old definitions that say that viruses are infectious particles. They are obligating intracellular parasite. Although this parasite definition, this parasite thing, is not quite right yet, because I don't know if virus fits to this concept of parasite, but you can say that. And viruses have genome with information to complete the infectious cycle, which I'm going to talk about later, what is the infectious cycle. And this one, virus, the definition, what is a virus? Virus is an organism with a capsid protecting a genetic material, a nucleic acid. So all viruses have capsid, and with that in mind, we have this proposal of Rayo in 2008. He proposed we doing this tree of life. He put virus and the ribosome encoding organism in this tree, all together in this tree of life. So it's a very nice proposal, because it probably considers virus as living things, and put in a tree of life that we can organize all these different kinds of life. And so they consider virus as capsid encoding organisms. And there are viruses from archaea, from bacteria, from eukarya, this is in the top. And in the bottom we have the ribosome encoding organisms, that are the actual living things that, as we know now, and there are bacteria, archaea, and eukarya, the three domains of life. And here we have the viruses as properly, and we have different kinds of viruses. We have this bacteriophage one, which has a capsid. A capsid is all of these, a complex capsid with a head, with a tail, with tail fibers. Its genetic material is DNA. We have this rotavirus. It's another kind of virus. It's an RNA virus, and it has some icosahedral symmetry in this capsid. We have this popular one. It's the HIV. And HIV is a retrovirus from the family retrovirus. And it is a RNA virus, and we have this one. Everyone have catched it sometime in his life. It's the influenza virus. It's a orthomyxovirus, and it's a RNA virus too. With the envelope, we have this other one here. It's a herpes virus. They are very large viruses with DNA genomes, and we have these adenovirus that which have icosahedral symmetry with these complex structures in the capsid. So it's adenovirus DNA virus. Here I show you a picture. It was published in Cell magazine in 2002. It's from the dengue virus. It's a flavivirus, and you can see here a very nice picture of it. It's probably icosahedral symmetry. And here, the most frightening of all right now is the Ebola virus. It's a filovirus from the family Filovirus. Its symmetry is helical symmetry, the capsid. Then I'm going to tell you that viruses are everywhere. And when I say everywhere, it's almost all the environments in the earth there are viruses. And in all the living organisms, there are some viruses that infect these organisms. And this is quite right, because we have no report of an organism which there are no viruses. So they are everywhere. And we can have virus in bacteria. In archaea is this microorganism that living in hot spring at high temperature. There are viruses infecting them. We have viruses in worms, in the slug in your garden. There are viruses in this cute giraffe. There are viruses in this funny lemur. It's a primate, very very close to us phylogenetically. And even the two most intelligent organisms in this planet, the dolphin and the mouse, they have viruses infecting them too. Here I show you that this estimation that there are at least 10 different viruses to each bacterial species. And here I show you a picture of my own. You can see here a Pseudomonas aeruginosa culture with these little plaques. and this little place is a phage from Pseudomonas, and I show you this part of the NCBI viral genomes page, and we have here a lot of Pseudomonas phages with genome complete, and just a part of them because there are a lot more of Pseudomonas phage that it couldn't fit here. And just to give you an idea that there are a lot of phages for just perhaps one genus of species, or some just for one species. And then they are abundant, you know. There are this estimation for Berg et al. In 1989, and he's estimated that there are 2.5 times 10 to 8 virus particles per milliliter of natural water. So that is just check on the number. It's kind of 100 million of virus particles in each milliliter of natural water in the ocean. So it's a lot. And here I bring you some graphs, some pizza graphs. In this graph you can see if you have all the, or if you catch all the living organisms in the ocean and you weight you weigh them, you have this biomass estimation, and that most of them are prokaryotes, are bacteria and archaea, and just this little of virus. But if you measure the abundance of these organisms, you find that there are much more virus than prokaryotes or protists or all the organisms together. So it's a lot. Here I bring you this concept about a virion and a virus, and I tell you that viruses are organisms with two phases. So there is this virion concept that says that the particle, the protein capsid with the genetic material, they are inanimated, they are a particle, and they are called virion. And when a virus infects a cell and it get in the cell and start to do some metabolism, some transcription, translating, and to replicate their genome, they are called virals. So there is this different concept. I'm going to try to use this correctly. The virion and the virus concept, they are two things different. And here I start to show you the structure and that virus can have. They can be their capsid, they are classified according with their capsid symmetry. So they can be helical, they can be icosahedral, or they can be complex. The helical capsid is this one. This is exemplified here by the Mosaco tobacco virus. It was the first one to be discovered and crystallized. This is an electron micrography of this virus. And you can see here that it's a spicle, it's kind of a spicle with 300 nanometers. And you can see here that there are just one kind of protein, one subunit of protein that they arrange in a way that produce this helical capsid. And you have inside this capsid the coded genetic material that can be RNA or DNA, as I'm going to tell you later. And we can have icosahedral capsid, the icosahedral symmetry, and here is exemplified by this adenovirus. And you have in the middle a cartoon with, you can see the icosahedral symmetry with this triangular faces. And you can see here in the electron microscopy this. Actually, you can see this icosahedral symmetry, and here we have a scan, a cartoon. You can see the spikes of this virus, and actually it has the icosahedral symmetry, but because of this spike and these different proteins, it will be considered a complex virus, but I'm going to tell you later. And here we have a computer assisted reconstruction of a rotavirus particle. Rotavirus is the virus that can give you some diarrhea, and when you go to the doctor don't know what you have. They say that you have a virus. Probably it was caused by rotavirus. Here in the top you have their electron microscopy, and you can see actually their symmetry. And you have the complex capsid symmetry, that they are the largest viruses, and you have the bacteriophagy T4 as a very good example of this. You have distinct components in the capsid with different symmetries, like here you have this icosahedral symmetry in the head, but not in the tail. And you have presence of different proteins devoted to special roles, like this tail and this base plate that is responsible for injecting the genetic material, and this head to enclose the genetic material. So these are bacteriophages. And here is all this symmetry, these three kinds of symmetry. The icosahedral, usually the virus that has this icosahedral symmetry, they can have an envelope, and this envelope is kind of a membrane made of lipids, and it is involving the capsid. So here you have herpes virus, an example from the family of herpes virus, and here you can see the capsid, the genetic material, and outside it you have this membrane, it's called envelope. And this microscopy, electron micrography, you can see actually the envelope. It's a very nice one. Here I show you the HIV, the very popular one. And you can see here the capsid, you can see the two molecules of RNA that are the genetic material. You can see this matrix, it's made of proteins to protect the capsid, and you can see this envelope outside of it with this protein, this receptor that are responsible for interacting to ligate with the cell that they will get in, they will infect, like the lymphocyte T1, T4, in the case of HIV. Here I'm going to talk about the genomes and classification are two things very related, because they use the type of genome and the strategy that they use to replicate and to produce mRNA to classify the virus. So virus can have DNA or RNA. They are the only organism known by now that can have RNA as genetic material. We know that RNA is very unstable, so it's different. And we have here two molecules of one of DNA, one of RNA, and you can see the difference. The T base, nitrogen base, nucleobase, is the difference between DNA and RNA, because RNA just can have uracil and DNA has T, thymine. And the sugar carbohydrate in this molecule is different too, because in DNA you can have the deoxyribose, and in RNA you can have the ribose. You just have the ribose in each one. We have here a lot of diversity about the genomes of virus, because they can be DNA or RNA, and this DNA can be double or single strand. We have these 24 families double strand that are the most usually ones, and we have these 5 families with single strand DNA, and we have this really different one is the hepatitis B virus family, and it has the double strand gapped DNA. And then if their genome are RNA, it can be single strand. There are 34 families. This single strand of RNA can be plus or minus strand. That plus, it means that the orientation of this molecule of the RNA is the same as the mRNA or the complement. So if it's the same, it's plus RNA. And they can be double-strand RNA. There are 8 families with that characteristic, with that genome. And besides that, their genome can be linear or circular, so a lot of trouble for us bioinformaticians to discover if they are circular or linear. And when you finally are able to assemble an entire genome, you have to discover if it is linear or circular, and they can have several fragments of on the genome, like the orthomyxovirus, the influenza virus. They have 8 fragments, so 8 different. It's kind of chromosome, but it's not chromosome because it has not the structure and the proteins linked to them to be called chromosome. But there are fragments of DNA or of RNA in this case. So here I show you the Baltimore system, and it's a very elegant system because it's mRNA centered, and it's elegant because just knowing the type of genome of a virus, you can infer their strategy to replicate and to produce the mRNA. So it's a very nice one. There are 7 types of viruses. The last one was added later because in that time Baltimore didn't know the hepatitis B virus type of genome replication, and then I put it in the top example of families that are in this class, just to get familiarized with that. So we have here the dsDNA is the most simple of them. Simple because it's usual for us. It's the same kind of replication and transcription that cells do. So we have the dsDNA, double-strand DNA, and just RNA polymerase DNA dependent can produce the mRNA. Just there is this enzyme in the cell, so just simple as it is. And you have the adenovirus as an example. But in the class 2 we have the parvovirus that are single-strand DNA. And this single-strand DNA isn't able to produce mRNA, so it needs to be double-strand, and this double-strand is accomplished by the DNA replication machinery from the cell. Can duplicate this single-strand DNA, and then this double-strand DNA can produce the mRNA. And here you have the 3 types of RNA viruses, the 3, 4, and 5. And it's important to keep in mind that in order an RNA virus can replicate their genome, it's needed an enzyme called RNA polymerase RNA dependent. It's a very important enzyme. There isn't this enzyme in the cell, so the virus needs to bring it with him this enzyme, and then to replicate. So we have here the double-strand RNA, and this double-strand RNA needs to produce mRNA. In order to do that, it will use RNA polymerase RNA dependent that it brings with him. It brings it with him. Yeah, he brings it with him. And so that's it. And you have the plus-stranded RNA, and this plus-stranded RNA, Professor Vêncio said a couple of times that it should win. It's kind of virus, the kind of genome in virus that should win, but just evolutionary talking. It's not scientific. It's not what happens, because all this diversity of genomes and viruses, they all are in the same bottom, are the same are successful equally. So this plus-stranded can get in the cell, and as soon as it gets in the cell, it can be translated by the ribosome for the cell and then produce the early proteins. So it's very important that. And for replication, it produces a minus-strand RNA, and this minus-strand RNA can produce by complementarity plus-stranded. So we have in the class 5 the influenza virus. There are a lot of nasty viruses in this class. There is the Ebola virus, and they are minus strand RNA. And in order to get in the mRNA, just simple, it needs to use RNA, polymerase RNA dependent that it brings with him. He brings with him, and this RNA polymerase RNA dependent, you produce the mRNA. And in the class 6, we have the retrovirus that are the HIV kind, and then they are single-strand RNA, but they are somehow different because the strategy involves a DNA intermediary. So the single-strand RNA is heterotranscripted to a DNA molecule. This DNA molecule will become a double-strand DNA, and this double-strand DNA will integrate in the genome, and it will become a provirus, and this provirus will then be transcribed in mRNA. It's a very different strategy. And you have in the end the class 7, and this class 7, guys, those are weird because they are double-strand DNA, and there is this RT in the end because it's kind of a heterovirus, but another way, because it is a double-strand DNA, a gapped double-strand DNA, and this gap needs to be filled up, and it is filled up, so the double-strand DNA gets completed, and then you would say, okay, now I just need to produce mRNA, but it's not. It produces mRNA or replication, sorry. For replication, just need to replicate this DNA to produce another DNA, the application of their genome. But they get RNA intermediated to produce this replicated genome. So it's very different. It's so just to make sure that you understand, it has a gapped double-strand DNA. This gap is filled up. This double-strand DNA produces a mRNA, and this mRNA is produces a cDNA, single strand. The single strand produces a double strand. This double strand is the genome replicated. So it passed for some unnecessary, in our view, intermediated, but we don't know why this happened. You just know that this really happened, and that is why they are weird. Is the hepatitis B virus. And here I show you the life cycle. I'm not talking deeply about this life cycle, but it's just important for us bioinformaticians to know that a virus can go in two ways to pass: the lytic cycle and the lysogenic cycle. In the lytic cycle, it infects the cell and uses all of its machinery to produce other particles, replicate their genome, and then it leaves the cell and release these virions in the ambient that will infect another bacteria. This is the lytic cycle. But interesting is that they can incorporate their genetic material in the genome of the bacteria or the genome of the cell and become a prophage or a provirus. And this prophage or provirus will reproduce within the cell, with the machinery of division from the cell, from application from the cell, and sometime in the life of the cell, it will some environmental triggers can make this virus become lytic again and then kill the cell and produce a lot of particles, and that is it. So it's interesting to look at this prophage or provirus thing, because when you are analyzing genomes, the sequence of genomes, you can find a way, you can build a way, an algorithm to try to find these prophages, these proviruses, and there are some couple of good software that try to do that. And that is very interesting to us to search, to find these virus, even the ones that are integrated in genomes, even the ones that are provirus. That's very interesting. And here I show you this special kind of virus. They are the bacteriophage. They are viruses that infect bacteria. They are the one that I work with, and they are the most abundant biological entities in the world. If you count, there in the ocean there are a lot of bacteriophages, and most of them have the double-strand DNA, and 96% of these bacteriophages are in the order called Virology. And they have perhaps an important ecological role that we are trying to assess now. We are just getting some information and some evidence that they might be important to control bacterial populations, and important in the carbon cycle. So it's they might be very important in the ecological. They might be very important in some microbial communities. So, and they are promising alternative to antibiotics, because there are a lot of bacteria getting resistant to antibiotics, and we are not producing a lot of different antibiotics, new antibiotics anymore. And then these bacteriophages, this phage, they perhaps, if you use them correctly, they can kill bacteria and then can do what antibiotics do to them now. And now I just finished this part, and this question, are viruses alive? And so we can explore it a little, and we've seen a lot of things in this seminar. We've seen that viruses have complex structures, that they have genetic material, that they replicate, and they have offspring, and they have this life cycle that can be leached or lysogenic. And they suffer selection. Of course they suffer selection, because they produce a lot of particles and they mute a lot. They have the diversity in the population, and this diversity will be selected, as Darwin said, by selection, natural selection. So this might be very good evidence that they are living organisms. Of course, that virions, if you consider virions, they are inanimated. So they are just particles like grain of sand, and they are just there. If you put a viral particle on your table, it will stay there until it gets on a cell, in a specific cell. Then it starts to do some metabolism in this thing, but it is inanimated while it is in your table. And I don't know, you just have to reflect about that. I have no answer to that too. And perhaps our concept of life should change, I don't know, because why just organisms that have this metabolism, that have metabolism all the time, they are considered life? Why not this particle can be considered life, or just, I don't know. It's a question, and I thought that would be interesting to explore it a little. And that's the end of the first part of my seminar. So this is the part 2 of my seminar. In this part I'm going to talk about metagenomics. I'm going to contextualize you to the metagenomic era, and just to give you an idea of how we can study viruses with the metagenomics approach. So in order to do that, I have to make some counterpoint to the genomic era. Then when you study a species, the species can be isolated, can be cultivated in a lab, for example. It's simple. It's kind of simple to study it, and you isolate this species, as Karen did here in the Pseudomonas culture, and you can see the phage here. And this phage is isolated, and then you extract its genetic material, obtain its genetic material. and then you use some reagents to build a library. This library has some fragments that have size that we know, more or less the size of this fragment, and this library is put in a sequencer machine, a next generation sequence machine, like a Illumina MySeq, for example. And this sequence machine will give us as output a lot of sequence of FASTQ file with a lot of sequence and their quality, and we use this sequence to assemble these reads, and we can assemble them, and if we are lucky we can assemble an entire genome, and then we can proceed with the annotation of this genome. In metagenomics, it's quite a little different because you have no isolated species. You don't need to cultivate this species in a lab. You can have some environmental sample from an ecological niche. For example, here you have the mouth of a baby, and you do some swab of his mouth, and then you have a community. It's a community. And you have several species. Here is represented by these different colors of different symbols. And it's a community composed of different species, different organisms. And you do a sequence, you do the same thing, you isolate its DNA, and you build a library, and then you put in a sequencer machine, and you get a FASTQ. Also, FASTQ is this FASTQ has fragments, a lot of reads that that came from different genomes. So you are not working with just one genome, but we are working with a lot of genomes, and you have these reads and this sequence all mixed. And it's the difficult thing, but because you have to assemble these reads, and it's not a easy thing, not really. And after you assemble these contigs, you can annotate these contigs. Sometimes you can obtain entire genomes from the metagenomic data, but that's not usual. That's not what always happens, and that is it. You can assemble different contigs that came from different genomes from an environmental sample. This is metagenomics, this is metagenomics. And why we are doing metagenomics? What's the purpose of that? And I tell you that most prokaryotes, we already know that most prokaryotes are not cultivated. So we can bring all the species that are living in the environment and bring to our lab and cultivate this, make some culture of this prokaryote from this bacteria. That's not possible. Most of prokaryotes are not cultivated, or we don't know how to cultivate them yet. That's it. So they are everywhere, they are in our communities, in microbial communities, and we don't know what they are doing, who they are, and that's the important thing, because they might have important roles in community ecology, and we want to know that. We want to assess this diversity. This genetic diversity is a lot of genetic diversity that we don't know, we know almost nothing about. And we want to assess this knowledge, and that's why we are doing metagenomics. And here I start to show you some projects, some metagenomic projects that these first ones are more general total metagenomic project, and this is a human gut microbial catalog that they try to catalog 3.3 million of genes, different genes, non-redundant genes from our microbial community our gut microbial community. So that was huge, because there are a lot of genes, a lot of proteins that we have no idea what they are doing, if they can harm us, or if they can do some good things to us. And these bacteria, this microbial community are producing these things, and we want to know. This is an effort to catalog these things. So it's a very nice project. And here we have this really nice project, it is the Earth Microbiome Project, and they are trying to coordinate a lot of samples that we are obtaining from all around the world. They are trying to coordinate that and study all these different environment, different microbiome that we are studying, put all them together, give some general information and specific information, but we are trying to study it to assess all the environments that are on Earth. And they are supposed to study 200,000 samples from metagenomics, from metatranscriptomics, and from amplicon of our ribosome RNA. Really nice project. And here we have this more specialized virus, and this is the Sorcerer 2 Global Expedition. It was they got samples from the oceans all around the world. They got samples from these places, all different places, and then they study. Of course, they study metagenomics in total metagenomics, but they study specifically the viral metagenomics, the virus in this community, in the samples from the ocean that they've got. And this is a very nice project. I recommend you to check on it if you want to know a little more about the virus in the ocean. And this is a Brazilian microbiome project that they are trying to coordinate an effort to get to coordinate the environmental samples and the metagenomic samples that we are having in Brazil. They said here in this article that Brazil hosts approximately 20% of the entire world's microorganism biological diversity, so that is a lot. They are trying to assess this knowledge, this genetic diversity through this program, through this project. And here is the Metazool project, the project which I work with, and we are studying environmental samples from 3 different microbial communities in the São Paulo Zoo Park. From these 3 microbial communities, from the howler monkey feces, from the lake, and from the composting. I work specifically with the composting data. I'm trying to, we have total metagenomic data, total metatranscriptomic data, and amplicon ribosome RNA data. And I'm trying to do some data mining and find the sequence from bacteriophages to be able to study the diversity and the abundance and perhaps their functional role in this microbial community, in this process. And we have already published one work in the +1, and which we analyze the diversity in 2 different composting units through high throughput sequencing. To our knowledge, it was the first work of its type, and we found some interesting results from the microbial community that associate to biomass degradation, and that we found that is mainly in the composting is mainly bacterial. And here I bring you some perspective that this is a nice magazine, the Current Opinion in Virology, and this article was published by Rosario and Berbert in 2011. And this is a overview of about what viral metagenomics can bring to us. What are the implications of the study of the viromes, this viral diversity through metagenomics? So you can see here that you study metagenomic, viral metagenomics, you can discover different new species. For example, you discover different species. You can solve evolutionary gaps that some gaps in the evolution theory and in the things about evolution that we do not understand. And this discovery of new species can solve these gaps. And if these gaps could give us a panorama, overview from the ecology itself, so it could give us information, data, to solve some ecology communities theories that we have not solved yet. We have no ideas of some things that can have some parasite host interactions, some AGT, that is the horizontal gene transfer, some of these things that we do not understand yet. So they could solve these problems, and the discovery could lead to discovery of new novel enzymes and annotation of no genes, and this could lead to some new enzyme that could be used in the industry. Like I just saw that in an article that they found in a virus from metagenomics sample, DNA polymerase, of course, that is unspecific, so it could replicate DNA or RNA with no specificity. And this is a very nice enzyme. I'm sure about that viruses are so different. There are a lot of things that are not common, and these things could lead to some nice thing in the industry, in some revolutionary thing like the PCR, the Taq polymerase enzyme could lead one day and give a Nobel Prize to the people that discover its application and all these things. And the discoveries of some new viruses could lead to surveillance, because some of these viruses could cause some disease, cause some pandemic or epidemic disease, and we know about these new species before they emerge as a pathogen or as a pandemic disease. A pandemic pathogen could allow us to do some surveillance, surveillance of this pathogen, and with one day it emerges, we are prepared to it. So that's the thing that you can do with viral metagenomics. And here I bring you a paper published in Current Opinion in Virology 2. It was by Mokili in 2012, and this is an overview of the viral metagenomics works published since 2002. Was the first marine viral metagenome, and since then we are doing a lot of work. The scientists are doing a lot of work. They are publishing a lot of viral metagenomics papers. You can see here in this graph the number of papers and how they are growing. And the new viruses, of course, the new virus and new enzymes from virus and new proteins, that they are fine because of these works, because you are studying, you are doing more studies, you are trying to assess the unknown, and these is allowing us to find these new things. So that is it. So this is the last part of my seminar, it's the part 3, and now I'm going to contextualize you with some bioinformatics applied to that knowledge, to that Coursera course, the knowledge that I have obtained from the Coursera course about virology. So this, I think it's important to understand your object of study and to do some good bioinformatics. So this is about the bioinformatics applied. And you can see here I'm going to talk about the challenges to study viruses, and you can see that I've already said that prokaryote culture, so most of them are, and either are viruses, because most of viruses are viruses from prokaryotes, and if this majority of prokaryote culture, either are viruses. And you said that, you can think about all the things that I've already said. You can think, oh, okay, so metagenomics is, seems quite suitable to study this uncultured majority, and actually it is. It's what we researchers are thinking, what we are doing to study viruses. And then you can do some metagenomics from a reef, from this ice in the North Pole, from this forest in the Amazon, and to study all viruses that are in these samples, in these environments. But and there are two ways you can study you can assess the viral diversity through metagenomics. One of them is you study total metagenomic studies that are total metagenomics. When I say that, I'm referring to a specific metagenomic samples, as you have all these bacteria, all this microbial community, and you do you sequence it, you do metagenome from it, and of course we will have virus in this community, but you can see the virus here, and this is one problem that we are facing, because there are from the output that we receive from the sequencer, a very few sequences are from virus, and it's kind of difficult to find this sequence and to understand if you are dealing with total community metagenomics. And of course you can do some effort to isolate all the viral genetic material in a sample, in an environmental sample, and then you sequence it. So you extract, you isolate just the viral DNA or viral RNA, and then you sequence it. And then we are going to deal with viral sequence, and we have not to do this effort to find this sequence in a ocean of other sequences. These are the two ways that you can study, you can do viral metagenomics. And talking about the first way that are total metagenomic studies, we have this problem that I've already said, that the viral sequences are very diluted. So to give you an example from a real work we are doing composting metagenomics, this is the result. You can ignore for now this no hit 73%, but you can see here that from the sequence that this software Kraken managed to identify, it was 4 million of sequence, and then these are about 26% of the sequence were identified. And you can see here a lot of bacteria species, a lot of different pseudoxanthomonas and such. But I call your attention for this number. Is the viruses sequence that Kraken identified, and is 0.03%. It's from the 4 million sequence, we have 1,230 sequence. It's very few sequence. What you are going to do with 1,000 sequence in a sample, from the environmental sample? So this is a problem that we are facing. And of course, just this sequence are related with a virus that we know, because this software, they do some similarity search, and these searches can provide us the information to say that this is a virus or this is not. But of course, there might be a lot of sequence from virus in this 73% that there were no hits. So to do that, with total metagenomic studies, we have to do some data mining, and this data mining involves a lot of algorithms, a lot of difficult algorithms that have to build to find this sequence, to find the viral sequence. And you can find this sequence by similarity or for some compositional methods. And it's a problem that we haven't solved it yet. It's a problem we are trying to deal with, and I'm doing that. It's kind of hard. And once we find the sequence, the viral sequence, we are going to do the assembly and analysis of diversity and abundance and functional notation, but only when we find the sequence. And it's a difficult thing, as I already said, they are very diluted, so that's the problem. And there's problems if you are dealing now with metaviromes. I'm saying metaviromes to talk about the purified viral particles metagenomics, that when you are not dealing with a lot of bacterial sequence, and then we have a huge amount of data. And this is a problem for all the metagenomic studies. If you are using next generation sequence, you have a million of reads usually, so that's something that we have to deal with. This sequence from the DNA, for example, and there are a million of reads, and this huge amount of data, they slow the processing. So it's difficult to do some similarity search. It takes sometimes too long. Taxonomic identification, and this is a work that I found published in 2012 by Hoes, and they studied two community lakes, and they purified the viral particles and then they sequenced them. And this is a study for virus, and you can see here the 73% of unknown sequence. From the null sequence, they use blast similarity to infer the taxonomic identification of the sequence. And then you can see here that even if purified viral particles metagenomic samples, they found a lot of bacteria sequence. And this is the virus sequence, but you have 40% of the identified sequence as viruses, and that's a lot, much more than the total metagenomic studies. And here you can see, but with this lining part, this shadow part, a sequence that had at least one viral sequence in the blast heap. So perhaps these proteins are all from virus, I don't know, they don't know. And here is a diversity analysis of the sequence that they identified as virus sequence. And here you can see that the microviridae family, that are small single-strand DNA, they are predominant in this sample in this layer. Here is you can use an approach based on commerce to proceed with the taxonomic identification. And this is a software that we are using here in the lab. It's the Kraken. And Kraken, it is study the commerce, the composition of commerce, and with this composition they do some heuristics, and this heuristic will allow it to infer the taxonomic identification. And you can, of course, do not use similarity to infer the taxonomic identification. So you can use composition methods based on composition of DNA. This is they are usually called signatures, and this is a paper published in 2006. It's about a classifier that here you can see one of their graphs, that you have the size of the sequence from 0 to 1000 and the percent of correct identification. And you can see it's a classifier based on a Markov chain. This is based only in the Bayesian probability. This is based in Markov chain, and this is based on a variable length Markov chain. They use commerce of number 4, And here you can see that the Markov chain based, it identifies more than 90% of fragments with 500 of size. So it's a very nice result. And talking about assemblies, so you have a virus, you have small genomes, you have no repeats, and it should be easy to assemble in the theory, but it is not. That's not what we are facing, because we have a lot of problems assembling these viral genomes, and sometimes you have very low coverage. And this is an example. So here you have a gap. In this gap, you will not assemble in a contig in an entire genome. So that's a problem. We have species that even if they are abundant, their genome are small. So this the number of reads you can find, the coverage is very small. So you have this problem. Sometimes you have very high coverage, but you have something like that. I had that one of my analysis, that we have the big contig, and I thought that it could be the genome, but we have in some parts of this contig you have the double of coverage in reads after I did some mapping, and I tried to identify the protein or the gene that was codified in this region, and I found it was a DNA polymerase. And it popped up in my mind that this could be a chimera. Chimera is the assembler are putting together reads from different species, from different genomes in one consensus. This concept is not it's not the flex of one genome, so it's a chimera. It's a amount of two or more species, two or more fragments from different species, from different genomes. And this happens because DNA polymerase is a very conservative protein, and they put all these reads together, and you can observe that there is some polymorphism, and this polymorphism are not desired, and this is a problem. You are assembling a chimera, a consensus. So we have these alternatives. We have all these assemblers. Mirror is a very good one. It's based in overlapping layout constants that they use the superposition of the reads to assemble it. It's a very good one. And we have SOAP de novo too. It's based on De Bruijn graphs, so it uses some heuristic to do the assembly. And we have this new one. It's very specific to viral populations, because it's supposed to identify the polymorphism, the difference in the viral sequence, and to do not put them together in a chimera, but mount and say to you that there is a polymorphism here in the population, and in order that you can identify this polymorphism. It's called vicuna. I haven't tried it yet, but I'm going very soon. And perhaps the ultimate challenge, the ultimate purpose of our bioinformatics analysis with metagenomics, metaviromes, is the functional annotation from the contigs that we assembled from this analysis. And there is some platform that can do that, they try to do that. Of course, it is not perfect, and we are learning a lot about that. We are studying how to do that. And there is this platform, the MGRAS. It's a platform to annotate and to do some quality controls, quality analysis in reads from these metagenomes. It's a very good one, we use it. And there is the IMG platform. It is supposed to analyze contigs that are the reads assembled in contigs. Then we can do some functional analysis more easier. and you can find orphs, you can find genes, you can find proteins, you can find orthologous groups in this context. And there is this one, this is kind of recent, and it's supposed to analyze reads as MD-RAS do, only faster than the MD-RAS, and it uses alignment-free methods. So I haven't used it already yet, but sometime soon I will use it. And for finish my seminar, I want to bring you this idea, this thing that we found, that most of the sequences are from the viromes, even the metagenomic ones, they are a vast dark matter. We know almost nothing about this sequence. We are trying to figure out their meaning by similarity searches or by other methods like the composition of DNA, but we have not been well succeeded about that. And this remains a dark matter. We know almost nothing about that. And you know here that Rosario Berbert in 2011 brought this study, and she found that 24 studies were published about viral communities, about viral metagenomics, and we now have this idea, we have this notion that these virus species, this viral diversity that we are finding now are very different from the ones that we know because they are well characterized, or the ones that we know. We have almost 2,000 viral genomes in the gene bank, but the ones that we are discovering now, they are really, really different. And this remains a dark matter. We know almost nothing, and we have like 810 megabases of data that we have no similarities hits to GenBank. It's a lot. And the analysis and retrieval and submission of this metagenomic viral data is difficult because they are not centralized, and even the ones that we can retrieve some similarity hits, they are really low similarity, less than 50%, so they might represent undescribed viral species. This is huge, guys. It's very important. We are trying to do with that. We're trying to discover and to assess this knowledge, but it's not easy. And all this new knowledge that we are having to do now has brought attention to the viruses. So metagenomics is a pop subject. People are doing a lot of metagenomics, and viral metagenomics are even more pop. And even we have to try to assess this knowledge about virus in a way, and perhaps you will be able very soon to do that. And because viruses are the reservoir of the genetic information of genetic diversity that we don't know yet, we want to assess the knowledge. That's the idea that I want to pass to you with my seminar, that viruses are very different. They have a lot of characteristics, and I wanted to know these characteristics and align this knowledge with the bioinformatics and try to assess this new knowledge in a way. I hope you have enjoyed the seminar. I'm sorry, because my English is not my native language, so I do not speak very well. And that is it. Thank you. I would like to thank Professor João Carlos Setúbal and Professor Aline Maria da Silva for being great advisors, the best. And I would like to thank Vincent Brancagnello for the excellent class that he put in Coursera. It's really good, really great. I recommend you to check on it if you want to know more about viruses. I would like to thank the guys from Citulalis for always providing helpful discussion, and I would like to thank Professor Ricardo Vêncio for realizing this discipline and this seminar and make it possible in all the ways. And like to thank you, my friends, for listening my exciting virus stories. At least I think they are exciting, but I don't know about them. But they hear me. They have heard me a lot. And thank you guys for watching it. Obrigado.